Hello everyone and welcome to our module on acute and chronic inflammation. In the video on inflammation principles, I talked about how there are two subtypes of inflammation. The first one is acute and the second is chronic inflammation. Acute inflammation generally has a rapid onset, such as minutes to hours, and it resolves quickly, usually within a couple of days. In contrast, chronic inflammation can last weeks, months, or even years. So in this video, we'll talk about some details of what makes up acute inflammation versus chronic, what cells are involved, and how the two processes play out. We'll start with acute inflammation, and it's very high yield to know that this is part of the innate immune system. In the immunology section, I talk about the difference between innate immunity and humoral immunity. Innate immunity is a non-specific response. It's a response of the body to infection or tissue damage that is the same for lots of different scenarios. And this is in contrast to humoral immunity, which involves B cells and T cells. It is a very specific response to one particular pathogen. There are three hallmark features of acute inflammation. The first one is increased blood flow. This is caused by vessel dilation. The second one is increased vascular permeability. And the third one is the emigration of neutrophils into tissues. These first two I talk about in detail in the video on inflammation principles. In this video, we'll talk a little bit about how neutrophils emigrate into tissues. And as I said on the last slide, acute inflammation, in contrast to chronic inflammation, has a rapid onset and short duration. Within a few minutes of a trigger, acute inflammation can begin, and it usually resolves within a few minutes, hours, or sometimes days. Shown on this slide are the elements of the innate immune system, which I discuss in the video in immunology. But what I want to mention here is a key element of innate immunity are phagocytes. These are capable of clearing debris, for example, damaged tissue. They can also clear microbes, and so they're very important in acute inflammation. You want these cells to consume any tissue debris, to consume any pathogens, and clear them from the body. The phagocytes in innate immunity are the macrophages and neutrophils. As we'll see in a minute, the macrophages can get the process of acute inflammation going, and then they call neutrophils into the site of inflammation, and neutrophils flood the area and become the dominant cells of acute inflammation. And all these other elements of innate immunity are also important, but the key ones are the phagocytes. So let's talk in detail about macrophages, because these are important cells for acute inflammation. These are referred to as the guardians of innate immunity. They're found in your tissues and they're capable of phagocytosis. And the reason they are called the guardians of innate immunity is because they are often the first cell to recognize either cellular damage or microbes or foreign bodies, and they will then go on to activate the acute inflammatory response. And macrophages aren't the only cells that do this. Similar roles are played by mast cells and dendritic cells, but we often talk about the macrophage as the prototype of the cell that can activate the innate immune response and trigger acute inflammation. The reason macrophages are the cells that can trigger acute inflammation is because they are able to recognize molecules that are foreign and that shouldn't be present in tissues. The two types of molecules they can recognize are called DAMPs and PAMPs. So DAMPs are damage-associated molecular patterns. These are molecules that are found in tissues only when there has been some form of tissue damage. For example, proteins in the mitochondria shouldn't normally be exposed in tissue beds, neither should DNA. So macrophages can recognize these, they're called DAMPs, and then they can trigger an acute inflammatory response. PAMPs are similar to DAMPs. They're called pathogen-associated molecular patterns. They're present on many microbes. They're not present on human cells, and macrophages can also recognize these. The way that macrophages recognize PAMPs and DAMPs is through something known as toll-like receptors. These are found on macrophages and also dendritic cells and also some other cells, including some epithelial cells. They're found on the cell membrane so that the macrophage can react to a PAMP or DAMP. They're also found in the endosomes. So if a macrophage phagocytoses something, it can then recognize if a PAMP or DAMP is present. These are referred to as pattern recognition receptors, and that's because they recognize the patterns found in PAMPs or DAMPs. When they recognize these, they secrete cytokines. They also become activated cells and send out more cytokines and other inflammatory signals, and this triggers the entire acute inflammatory response. And besides toll-like receptors, there are other activators of macrophages. The Fc portion of antibodies can activate macrophages, and complement proteins can as well. So toll-like receptors and PAMPs and DAMPs are one way that acute inflammation can be activated. A second way that acute inflammation can be triggered is through what's known as the inflammasome. An inflammasome is a protein complex. It's a big clump of proteins found in the cytosol of many cells, and it's very important for recognizing cell damage. Through mechanisms that are incompletely understood, the inflammasome can become activated by certain components of damaged cells. Some of the components that have been best described include uric acid, extracellular ATP, and free DNA. So if these things are floating around in tissue beds when they shouldn't be, the cell will recognize it through the inflammasome, and that will trigger acute inflammation. What happens is the inflammasome, when activated, leads to production of IL-1, and this is released, and that triggers the release of other inflammatory mediators from other cells, and thus through the inflammasome, anytime there's tissue damage, acute inflammation can be activated. Once acute inflammation begins, there are a number of so-called mediators of inflammation that are released by various cells, including macrophages and other cells, and these drive and sustain the entire acute inflammatory response. Some of these are the vasoactive amines. These are substances like histamine and serotonin. Other mediators are lipid products derived from arachidonic acid. These include things like prostaglandins and leukotrienes. And I talk about both vasoactive amines and lipid products in the video on inflammation principles. And then finally, complement is also an important inflammatory mediator, and I talk about that in the complement video in the immunology section. Now let's talk about neutrophils, because these are very important cells for acute inflammation. These cells come from the bone marrow and they circulate in the bloodstream for about 5 days, and then they die through apoptosis unless they become activated. If they are activated by some of those inflammatory mediators, then they can be drawn from the bloodstream to sites of inflammation, and once they get there, they can perform phagocytosis. So even though macrophages can perform phagocytosis, one of the most important things macrophages do actually is not phagocytosis, but it's to call in neutrophils. Neutrophils are like the on-call team for inflammation, and they will rush to tissue sites and provide extra support in terms of phagocytosis when there is dead tissue or some sort of pathogen. The process by which neutrophils exit the bloodstream to enter tissue beds where inflammation is occurring has been well described in the basic science literature. The first thing you should know is that neutrophils exit the vascular system at post-capillary venules. If you look at this cartoon on the screen, these are the capillaries here in the middle. This is a post-capillary venule. And in these venules, endothelial cells contract in the setting of inflammation. I talk about this in the video on inflammatory principles. When they contract, they open up gaps between the endothelial cells, and that's how neutrophils get into tissues where inflammation is ongoing. And there are 4 steps to the process of neutrophil extravasation, the exit of vessels into tissues. The 4 steps are called rolling, crawling, transmigration, and migration. Each of them is mediated by different binding between ligands on the neutrophils and molecules on endothelial cells. And I describe this entire process, including the 4 steps, in detail in the immunology video on innate immunity. Very high yield for you to know the typical timeline of acute inflammation. So in the first 2 days or so of acute inflammation, neutrophils are the dominant cell type. There are many of these in the bloodstream, and they quickly attach to the endothelium and migrate into the tissue beds where inflammation is going on. They then undergo apoptosis after about 24 or 48 hours because neutrophils in general do not live that long. After 2 days, the dominant cell type are monocytes and macrophages. These cells live much longer. They can also replicate in tissues, so they hang around after the neutrophils have gone away. So if you imagine someone who has, say, a skin infection or some other type of inflammatory condition, maybe they've suffered from trauma, and an inflammatory process is going on over 3 or 4 or 5 days, these are the cells that are going to dominate early and late. It's very high yield to know that for your exams. Here's a typical timeline of acute inflammation. You can find graphs like this in Robbins Pathology and many other textbooks. The first thing that happens in areas of inflammation are that edema occurs. That happens very rapidly within a few hours of a trigger for acute inflammation. Following that, neutrophils flood the area, and they are the dominant cell until about day 2. Now, in many cases, acute inflammation resolves by day 2, and so the neutrophils take care of the entire problem. However, if the process persists beyond about 2 days, the neutrophils begin to die, and macrophages and monocytes become the dominant cells. And that's why, as we'll talk about later, in chronic inflammation, which can last days, weeks, or months, you do not see neutrophils. Instead, you see monocytes and macrophages. Now, what I just showed you is the typical time frame for acute inflammation, but there are exceptions, and I've written a few of these on the slide here. Pseudomonas infection is famous for having neutrophils that dominate for days. In viral infections, lymphocytes are often the first cells to appear at the site of inflammation, and then in hypersensitivity reactions, as you may know, eosinophils dominate and play a very important role. Let's talk about the resolution of acute inflammation. There are 3 potential outcomes once acute inflammation begins. The first one is simply resolution of the process. So if phagocytes are able to remove all the microbes and debris, then the tissue bed will return to normal and the phagocytes will die off. Second possibility is that the tissue will heal or develop scar. This is what happens when the tissue damage is too extensive for regeneration and connective tissue has to grow to replace lost tissue. And I talk about healing and scar in a separate video. And then the third possibility is that the acute inflammation persists and now becomes chronic inflammation. So let's talk about that now. Chronic inflammation is prolonged inflammation that lasts for weeks or months. It can develop from acute inflammation, but it can also begin on its own. There are some inflammatory processes that begin very slowly. They are said to be smoldering, and they basically never have an acute phase. They go right into chronic inflammation. And one of the hallmarks of chronic inflammation, and one of the things that makes it distinct from acute inflammation, is that in chronic inflammation, you have tissue destruction from the inflammatory process and tissue repair occurring at the same time. And that's a key feature that defines chronic inflammation. So what are some of the potential causes of chronic inflammation? Well, there are some infections that are persistent. Certain microbes are very difficult for the immune system to clear. For example, mycobacteria, especially tuberculosis, often lead to chronic inflammation. There are also a number of parasites like helminths that are very difficult for the body to eradicate, and they can lead to chronic inflammation. And in these cases, what happens is you have a prolonged infection, and basically the inflammatory response turns in to a version of a type 4 hypersensitivity reaction where you just have chronic cell-mediated immunity trying to remove these types of infections. Many autoimmune diseases result in chronic inflammation, and then finally prolonged exposure to substances that the body has difficulty clearing can lead to chronic inflammation. So silica, when it gets into the lungs in certain industrial workers, can lead to chronic inflammation. And then the underlying cause of coronary artery disease and other vascular disease is believed to be cholesterol, which drives a chronic inflammatory process. The main cells in chronic inflammation are mononuclear cells like macrophages, lymphocytes like T and B cells, and plasma cells. You do not see neutrophils in chronic inflammation in general. Macrophages are the dominant cell type. They secrete cytokines and they also activate the T cell response in chronic inflammation. And it turns out that the activated macrophages seen in chronic inflammation come in two forms. Some are called M1 macrophages. These have been activated via the classical pathway to destroy microbes. I'll talk about what this is in a minute. The second type are the M2 type. These are activated via what's known as the alternative pathway, and their role is mainly tissue repair, not destruction of microbes. Just like there is a classical and alternative pathway for activation of the complement system, there is also a classical and alternative pathway for activation of macrophages. Let's talk first about the classical pathway. This generates so-called M1 macrophages. There are a number of ways this pathway of macrophage activation can be triggered, but one of the classic ones is by a microbe. For example, endotoxin found in many microbes can activate toll-like receptors on macrophages. Once this type of macrophage becomes activated, it stimulates T cells, and then T cells stimulate macrophages. Basically, chronic inflammation is a lot of crosstalk between T cells and macrophages. The T cells are very important. And what they do to drive classical activation of macrophages is T cells release I, F, and gamma. This is very important for activating the M1 macrophages. And an easy way to remember this is that there is one G in macrophage, and it stands for I, F, and gamma. Once macrophages are activated via the classical pathway, they respond by generating reactive oxygen species. These can be used to kill microbes. They also synthesize more lysosomal enzymes to kill microbes, and they also secrete cytokines which drive inflammation. And because they are driving inflammation and drawing cells into areas of inflammation and releasing toxic substances to kill microbes, they can destroy tissue, and therefore the M1 macrophages are responsible for much of the tissue destruction that occurs at sites of chronic inflammation. The alternative pathway of macrophage activation leads to so-called M2 macrophages. And this pathway involves macrophages being activated by something other than interferon gamma, some other cytokine. So besides interferon gamma, many other cytokines are produced as part of the inflammatory response. And some cytokines produced by T cells, especially IL-4 and IL-13, can also activate macrophages just like interferon gamma, but they don't generate the same response. They generate an alternative response. And what activated macrophages via the alternative pathway, the M2 macrophages, do is something different than the M1 macrophages. These macrophages actually inhibit the classical activation of other macrophages, and their main role is tissue repair. They secrete growth factors, which leads to angiogenesis and tissue repair. So what you've got going on at a site of chronic inflammation is some M1 macrophages, which are actually destroying tissue and trying to destroy pathogens, and also M2 macrophages, which are trying to repair tissue. There's a couple of potential outcomes of chronic inflammation. One is that it can lead to scarring. So, for example, patients who have hepatitis B infection of the liver that is chronic will have chronic liver inflammation, and that can lead to cirrhosis because scar tissue is laid down in place of healthy liver tissue. Chronic inflammation can lead to secondary amyloidosis. I talk about amyloidosis in a video on its own in the hematology section, but any chronic inflammatory process can potentially produce proteins that form amyloid, and this can lead to the secondary forms of amyloidosis. And then finally, chronic inflammation is a risk factor for malignancy. Anywhere that chronic inflammation is going on, there's lots of cell stimulation and growth. And sometimes this can stimulate a cell to such a degree that it becomes overactive and begins replicating uncontrollably, and that's malignancy. And in a way, this is similar to hyperplasia. Hyperplasia can lead to malignancy. The same is true of chronic inflammation. It can lead, like hyperplasia, to dysplasia and ultimately neoplasia. And there are many, many examples of chronic inflammation leading to cancer. Chronic hepatitis can lead to liver cancer. H. pylori infection of the stomach is a chronic inflammatory process that can lead to gastric cancer. There are many, many other examples. I'll finish with this slide and just give you an example of two tissue beds undergoing acute versus chronic inflammation. So on the left is a tissue bed undergoing acute inflammation. There are lots of neutrophils, which are the cells shown on that slide with multi-lobed nuclei. This is one up close at the top. On the right side of the screen is a tissue bed undergoing chronic inflammation. You can see that there are no multi-lobed nuclei like you would see with neutrophils. Instead, all you see are mononuclear cells with a single round nuclei, and that's because the monocyte macrophage shown at the top of the screen is the hallmark cell of chronic inflammation. And that concludes our video on acute and chronic inflammation.